I think we're going to get started. Are we okay to get started? Yeah? Okay, good. Okay, welcome everybody to our last presentation of the 2015 CCOVI Lecture Series. Today we are pleased to welcome Wendy McFadden-Smith, who is a CCOVI professional affiliate and the Tenderfruit and Grape and Integrated Pest Management Specialist at the Ontario Ministry of Agriculture, Food and Rural Affairs. Her research focuses on etiology of sour rot, grape fungal pathology, grape berry moth management, and integrated pest management. She is also a sessional lecturer in the Department of Biological Sciences here at Brock University, as well as a lecturer at Niagara College. Examples of her research projects include integrated management of grapevine powdery mildew and impact of powdery mildew on quality parameters of grape juice and wine. So please join me in welcoming Wendy McFadden-Smith.
Thank you for coming on a day when we finally have sun and warmth. I'd much rather be outside pruning grapes too, but let's get this started. So my topic is viruses, vectors, and vines, oh my. This is a team project, several people involved, myself, Dr. Lauren Stobbs and Christelle Olivier with Agriculture Canada in Vineland and eventually London. Neva Grigg is a technician at Ag Canada. Julie Lupia did some of the work with mealybug vectors as part of her undergrad thesis project. And Catherine Hoshku and Tafik Kabaj, I hope I said that right, were consultants who helped with sample collection and analysis. So in August of 2013, we started seeing some symptoms that look like this. Red varieties tend to turn red if you look at them wrong, so we weren't too worried. A little bit more purpling in some varieties, some kind of weird interveinal reddening on other varieties. Getting into September, some of this marginal discoloration started to turn red. We also saw some kind of weird symptoms with red blotching and red veins. Different varieties had different symptoms, but the symptoms were a little bit different in some vineyards than in others. And we did see symptoms in whites as well, this interveinal chlorosis. In some cases, the distribution was very scattered, individual vines here and there showing these symptoms. Side by side, you might have seen a vine that was expressing these symptoms beside one that was completely symptomless. In some cases, these were grouped together in patches. You might see sections of panels that were infected and then no symptoms. So my response, it's potassium deficiency. Boy, was I wrong. What we were seeing widespread were symptoms of grape leaf roll. Now, we knew grape leaf roll was present in Niagara, but the assumption was that it was in relatively low numbers based on surveys that had been done in the past. The symptoms of grape leaf roll tend to start in late August to early September, just as we're getting past veraison into the full coloring of the fruit. Symptoms start from the base of the shoot and progress upwards into the younger leaves. Very typical of these symptoms, downward curling of the leaf margins and interveinal reddening. So the veins stay green, the interveinal areas turn red, or in the case of white varieties, they may turn yellow. So these are actually classic symptoms. We were out doing a survey in one vineyard that had quite extensive infections, and actually ran across photographers who were taking color pictures of the beautiful fall foliage in the vineyard. Classic symptoms in a Chardonnay vine, the downward rolling, very characteristic. So this meant that I had to do a crash course in grape leaf roll and other diseases, viral diseases. So digging around, getting some information from a variety of different colleagues, Mark Fuchs being one of them. This is an example of the effects of leaf roll infections on Pinot Noir. So you'll notice that the fruit is these were collected at the same time. The fruit is definitely delayed in maturity, has lower Brix, you can't tell that looking at it, but it does, and reduced anthocyanins. Some of the work that was done by Martinson and the group in the Finger Lakes looked at the accumulation of Brix in infected Cab Franc versus healthy, ones that were asymptomatic, that were free of the virus. And you can see consistently throughout the growing season, the leaf roll infected vines were delayed. The Brix was significantly lower. They also did analysis of infected, that they were tested for the presence of the virus, infected vines versus uninfected, and this graph shows the degree of Brix reduction that occurs in infected vines relative to uninfected. So anywhere up to as much as 3.5 degrees Brix reduction in different vineyards that are infected with leaf roll. So these would be infected vines compared to uninfected. The symptom, the reduction is variable. I'm not sure whether they actually quantified the amount of virus expression, why there's so much variability, but who can afford reduced Brix in their Cab Franc anyway? Reduced anthocyanins, wines made from grapevine, leaf roll, infected virus 3 versus healthy. You can definitely see a difference in the color. So grapevine leaf roll associated viruses, it's a complex. There are actually, the literature lists 10 different leaf roll viruses that can cause these symptoms. The two predominant ones are leaf roll 3 and leaf roll 1 internationally. The other ones occur sporadically in different areas. Leaf roll viruses will infect all the vitis. So vinifera, labrusca, the French hybrids, rootstocks, everything is susceptible, can be infected by leaf roll viruses. What's tricky about leaf roll viruses is that the symptom expression can be absent or very variable. The virus, the strain, the virus, whether it's 1, 2, 3, etc., the strain within that virus, whether there are mixed infections of multiple strains, other viruses, these can all affect the symptom expression. The rootstocks can affect it. The varieties, certain varieties can be infected and be completely asymptomatic. The season and environmental conditions can affect symptom expression and production practices, so especially crop load, leaf removal, vine balance, very important. So just looking at things that look like, sort of like leaf roll but aren't, this is taken from a slide that Tom Lowry from Ag Canada and Summerlin put together. Classic leaf roll symptoms with the interveinal reddening, the green veins, downward cupping leaves. This one, they don't know what it is, Syrah decline, and zinc deficiency. So there are a lot of things that could look like leaf roll that aren't. It can be spread by propagation of infected vines. As I said, vines can be infected but asymptomatic. So nurseries may collect budwood or rootstock material from infected vines, not knowing that they were infected. There are insect vectors that can transmit leaf roll virus, but there's no mechanical transmission, and this is important. It cannot be transmitted by pruning, going from an infected vine, pruning to an uninfected vine, by hedging, trimming, harvesting, suckering. None of these operations will allow the transfer of grapevine leaf roll virus from infected to uninfected vines. So that's good news. Gotta be some good news. So the insect vectors of leaf roll virus have been identified as mealybugs and soft scales. These are sap-sucking, so phloem-feeding insects. There is no specificity, so a number of different mealybug species have been shown to be vectors of leaf roll virus, specifically leaf roll virus 3. The virus is semi-persistent, so it will remain within a growth stage of the insect, but not be passed on when it moves into the next growth stage. And feeding by a single insect that's carrying the virus can result in infection of a whole vine. These are the insect vectors of grape leaf roll associated virus that have been identified in Canada, or that have been identified. The ones that are highlighted in red are species that have been found in Canada. Work in the Finger Lakes has identified cottony maple scale, European fruit licanium scale, and great mealybug to be the predominant vector species present. Just briefly, because I'm a plant pathologist and I love disease and life cycles, an overview of a life cycle of a great mealybug. This is the adult female, this is the adult male. Only the males have wings, the females are sessile. They will go through a number of instar stages. Each one is very similar to the next in general morphology. Our observation has been that the younger ones tend to be a brighter, kind of a salmony pink as far as grape mealybug is concerned. And as they continue in their development, they will become very white, covered with waxy scales. So this is the male development, this is the female development. Great mealybugs are much more efficient at transmitting leaf roll virus than the soft scales. And the earlier instars, so the younger growth stages, are more efficient at transferring leaf roll virus than the more mature stages. Here's an example of some transmission work that was done so you can see how tiny these first instars are. This is a leaf disc that was taken from a vine on which great mealybug had been feeding. It was infected with leaf roll virus. These insects and the leaf disc were transferred to a healthy vine to show transmission. So what was found in these greenhouse transmission studies was that the mealybugs were placed on the infected vine for 24 hours and then transferred to healthy vines. And within 3 months, 40% of those inoculated vines showed infection. This group also did a survey looking at which viruses were being carried by these different vectors. So they looked at 75 different mealybug individuals, 24 different soft scales, and actually used ELISA or PCR, I can't remember which, to determine whether they were carrying a strain of grape leaf roll. And what they found was that the mealybugs tend to be carrying a lot larger proportion of the mealybugs tended to be carrying the leaf roll virus than the soft scales. This is taken from a presentation that was provided by Monica Cooper at UC Davis. She also spoke at Ontario Fruit and Veg this past February. They did some similar studies where they did inoculations with transferring mealybugs from infected vines to uninfected vines on July 14th. They looked for symptoms for the rest of the season, did sampling and analysis with PCR to determine whether the vines were infected, and then watched for symptoms in the following 2 years. What they found was that in the inoculated vines, there were no symptoms the first year, but by 2012, the year following, 29 out of 60 of the vines were showing symptoms. So there's a delay in the expression of symptoms. So the first instar nymphs are the most efficient vectors. They are also the most mobile. They move the most. The older, it's kind of like us, the older we get, the less we want to move around. They may walk from vine to vine, so there may be some movement of the virus from one vine to an adjacent vine. Studies have shown that they can move up to about 1 meter walking. They can be windborne. Groswitz and James showed in their sampling that a very low proportion could be found up to 8 meters from the vine that had been, on which mealybugs had been placed. So relatively low movement as far as wind is concerned. This is a figure from The north coast on the Pacific Northwest showing the development of grape mealybug relative to vine development. So it overwinters as first and second instars under the bark. Very important as far as control is concerned. Under the bark. Very difficult to get anything that's living under the bark. As the temperatures increase, the nymphs will move out onto young developing leaves. In June, the females will move back out down to the bark, lay their eggs, and then in late July, some nymphs will move out, and in late August, the females will return. So the movement is relatively localized, generally around protected areas. Now, one of the things that's different is that we very seldom see grape mealybug on the fruit here. Very, very low frequency that we'll actually see it. This is some work by Tom Lowry in BC, where they were monitoring Cab Franc infected with grape leaf roll. Here, 2011, pink means that the vines were showing symptoms. Blue, light blue here, infrequently. These are vines that were kind of iffy. Green, asymptomatic vines. 2012, symptom expression was not as significant. It was a lot harder to pick out those infected vines. But when they went out in 2013, look at the change. The number of vines with pink. Large increase. And focal movement, if you tried to relate these to over here, the movement from those pockets of infected vines down the row. The other complicating factor, red blotch. This is another viral disease first recognized in 2008 in Napa. It's not a new disease. It's just been recognized as a specific viral disease. It's actually been identified in archived material of vinifera early Burgundy that was collected in Sonoma in 1940. So it's been around for a long time. It's just not been recognized as red blotch disease. Like leaf roll, the symptoms first appear in late August to early September. These symptoms, irregular pink to crimson blotches on the blades, once again starting at the basal leaves and progressing up to the younger leaves. Secondary and tertiary veins will turn red. So generally you'll see this developing from the margins of the leaves in. But this is inconsistent. Not necessarily will you see the red veins in a positively infected vine. Occasionally, the intervenal areas between secondary veins will turn red, but not all the time. The big difference between red blotch and leaf roll, no downward cupping of the leaves. Some examples from Mark of red blotch symptoms, the blotching between the veins, across the veins, not as distinctively intervenal as we see in leaf roll. So why didn't we know it was a viral disease? It was misdiagnosed as leaf roll historically before 2008. However, when those vines were tested, didn't come back positive for leaf roll virus. That's because red blotch is caused by a DNA virus and leaf roll is caused by an RNA virus. So the tests that we would use for leaf roll would not detect red blotch. Since it's been identified, it's been found in a number of US states and also in Ontario and BC. It's been found on a variety of different cultivars, some red, some white. Not all of these are grown in all of these locations. This is some work that Monica presented back in February, looking at incidences of the different virus diseases in vineyards that have different planting dates. So going back as far as 1880, red blotch in 0.4%. And pretty comparable with across from old to younger plantings. If anything, these newer plantings are cleaner because we now have the technology to be able to test for infection to try to reduce planting of infected vines. but just significant that even vineyards that were planted in the late 1800s were infected. Impacts of red blotch on fruit, lower Brix, higher malic acid, no consistent impact on titratable acidity. In trials that were conducted in California, reducing crop load did not consistently improve berry composition or Jur's chemistry. No matter what you do, there's going to be a typo. And there's no clear impact on yield. This is some of the work that's been done in California. These are red blotch infected, probably reds and whites. Fruit from red blotch infected, decreased anthocyanin, decreased tannin, decreased phenols, total anthocyanin, tannin, phenols, significant reductions in these. Quality tasting notes, wines made, Cabernet wines made from red blotch infected grapes, described as green, unripe tannins, lack of finish or length, hole in the middle palate, and less texture. Not just the quantity of tannin is affected, but the quality as well. And they're continuing to look at differences between infected and non-infected vines. Vectors of red blotch. There was a Washington study a while ago that suggested that Virginia creeper leafhopper may act as a vector. Now this made people in most grape growing areas very nervous because Virginia creeper is a very common pest species in vineyards. So if it has the potential to actually transmit this virus, we could be in trouble. So some work that was done in California looking at screening different grape pest species for transmissibility of grape red blotch virus. So once again, they took groups of infected vines that had red blotch, put the potential vector species on those, left them for up to 72 hours, then transmitted those or moved those vectors to healthy vines and monitored them for the presence of red blotch. None of those vectors tested have been able to transmit red blotch. Yay! Once in a while I get to give good news. So, this big explosion of red vines that we saw in 2013 got us thinking maybe we should be looking at what's going on in Ontario. So, with funding from the Grape Growers of Ontario, we designed a survey to look at the incidence of leaf roll virus and red blotch virus in Niagara vineyards. In 2013, we focused on red varieties of vinifera, and in 2014, we homed in on Riesling and Chardonnay. In 2013, we looked at 20 commercial blocks for leaf roll virus and 20 blocks for both leaf roll and red blotch. We determined the number of samples based on the acreage in each sub-appellation in Niagara. Got to be politically correct. So, in areas where there was a higher acreage of red varieties grown, red vinifera varieties grown, the number of samples dedicated to that particular region would be higher. It took a lot of training to convince this consultant not to be swayed by the growers. We were provided with maps of the farms and randomly selected the blocks that we were going to sample. Because obviously a grower is going to want you to try and sample the ones that he knows or she knows have symptoms or they think have red blotch or leaf roll. And we wanted an overall picture of the incidence. So we didn't want to bias it toward infected vineyards. So within each grower location, the block was randomly chosen and the minimum size was to allow a 20-panel sample. So this map shows the different sub-appellations and the sampling intensity. I know the numbers look really small, but this was a very rapidly assembled and not extensively funded project, so we had to work as efficiently as possible. So the white numbers show the number of vineyards that were sampled for both leaf roll and red blotch, and the ones in black show the number of vineyards that were sampled for just leaf roll. With this random sampling, we ended up collecting samples from 21 Cab Franc, 4 Cab Sauv, 6 Merlot, 2 Pinot Noir, and 1 Gamay block in Niagara. The survey was localized to Niagara once again because it was about a 3-week period from when we got confirmation that we had the funding till frost. So it was a pretty small window. In 2014, we were able to acquire funding to test for both leaf roll virus and grape red blotch virus in white varieties and the distribution of the different numbers of vineyards sampled in the different sub-appellations here. In this case, we were sampled 14 Chardonnay and 16 Riesling blocks. The sampling pattern was based on advice from Mark Fuchs, where we took 20 samples from each vineyard. Each one of these red squares would represent one post length, one panel. So 5, 6, 7 vines, whatever is between 2 posts in a vineyard. And 3 leaves from each vine were collected into a composite sample, and then the virus was analyzed from that composite sample. So we couldn't identify individual vines that were infected, but we could identify areas or panels within the vineyard that were infected. So basically, sample this panel, skip 4, sample this panel, skip 4, and so on. Skip 4 rows, sample, skip 4 panels, sample, etc. So we sampled 20 samples per block. We did either 3 or 6 basal symptomatic leaves from each of the 5 vines. 3 if we were analyzing for just leaf roll, 6 if we were analyzing for both leaf roll and red blotch. Leaf roll virus was tested at Ag Canada, and red blotch virus was tested at the Pest Diagnostic Center. We used ELISA kits to test for grape leaf roll virus and PCR to test for red blotch. This is a type of report that was provided to the growers. So they had a map of their vineyard. We showed where we entered, so they could actually identify which panels we had sampled that came back as positive or negative for leaf roll and red blotch. Overall, looking at Cabernet Franc, this shows the incidence percentage of panels. So this is not over the whole vineyard. This is of those 20 panels that were sampled. Very important. Of those 20 panels that were sampled, the proportion or the percentage of panels that were infected with leaf roll. So you can see that it ranged anywhere from completely free of leaf roll up to 100% of the panels infected. Then we add to this red blotch. So this poor grower thought they were getting off really well as far as leaf roll was concerned, then found out that they were hammered with red blotch. Same with this one. So the two aren't necessarily associated. You can have red blotch without leaf roll, you can have leaf roll without red blotch, you can have both of them together. Other varieties, not so much red blotch. Only one of the blocks that wasn't Cab Franc actually showed up with red blotch. But once again, a lot of variability in the incidence of leaf roll within the different blocks. Summarizing this, to specify, the kit that we used was a combination for grapevine leaf roll 1 and 3. So we don't know whether this is leaf roll 1 or whether it's leaf roll 3. This was just to be economic. Both of these viruses can be spread by mealybugs, so we figured that was relevant. We started out just checking for 1 and 3, but then we had leaves that were showing symptoms that were coming up as negative. So we expanded this to include leaf roll virus 2 as well. So what we have here are out of the 40 blocks sampled, 12 of the 40 had no infection, and this is just showing the relative incidence. So when we get into the red, the hotter the color, the higher the incidence of virus. Red blotch, 1, 2, 4, 5 out of 20 were positive. So definitely not as significantly distributed as the leaf roll virus. This just shows whether leaf roll 1 and 3 or 2 were present within a particular block. So if it's the red shows leaf roll virus 1 and 3 only, the yellow leaf roll 2, and purple leaf roll 1 and 3. So the number of vines or number of panels within each location that had combinations or solo infections. So once again, all over the board, but leaf roll 1 plus 3 being the predominant one. The other thing that we did, which was kind of fun, was we rode around on golf carts and called out positive, negative, positive, negative, positive, negative. How many vines are there in an acre of grapevines? Anyway, lots. So what this represents is a spreadsheet where red were positive, green were maybe, or young vines that were replants, and green was no infection. So we had several vineyards that we did this kind of representation, just to get a feel for the distribution. So this was done in September of 2013. In March of 2014, because we could identify where we started, we knew exactly which panel was which, we went back and actually sampled from positive and negative vines. So 20 vines were sampled, 10 that were positive for leaf roll symptoms, 10 that were negative for leaf roll symptoms. From each of these vines, we tested canes, so cane beetle tissue was collected, and tested for leaf roll 1, 3, and 2, and also for red blotch. And the other thing that was done was on those same vines, we collected 2 canes per vine, 10 buds per cane, and did cold or winter hardiness, cold survival in March. So, I'm sorry for people who are colorblind, but red is positive or was symptomatic, green was asymptomatic. So this was row 18, vine 1, vine 3, vine 6, and so on. Testing these molecularly for the presence of disease. None of them were positive for red blotch. None of them were positive in this vineyard for leaf roll 2. And interestingly, or encouragingly, only the vines that were symptomatic came up as positive for leaf roll 1 plus 3. The other thing that's really interesting is there was no significant difference in winter hardiness between vines that were infected and vines that weren't. Now the literature extrapolates that, and your assumption would be that because the green area, the photosynthesizing area is reduced because it's turned red, that there'd be less sugar to help with winter hardiness. But this isn't what we saw. The average percent viable buds with infected vines was 60%, uninfected 57.5%. Now in talking to some growers at this past year, the observation is that vines that had leaf roll tend to not have a severe cold injury. So perhaps leaf roll infection is a control for cold injury, or not. In the 2014 sample, dark green is Chardonnay, showing once again the incidence of leaf roll 1 plus 3. A lot of variability. Riesling, quite a bit less. One sample out of the 600 samples that were analyzed came back positive for leaf roll 2, and none of them came back positive for red blotch. So, we know that it's here. What are we going to do about it? So one of the things that we decided to look at as part of Julie's thesis project was to find out what vectors of leaf roll virus are here, and then there's another mistake. Try to relate development of the vectors to temperature and growth stage of the vines. So here's Julie. She actually did some work that year. This was, she's not watching, she's in Italy, so it's safe. Based on some of the suggested methodology from California, we did timed counts, the royal we, 4 minutes per vine, 10 random vines per site. So to do this, the students peeled back the bark and looked underneath the bark for the presence of mealybugs and scale. For each vine, they recorded the number of egg clusters, the number of first instars. So this would be a first instar here, and you can see the gradual increase in size as they go through. Second and third instars in adults, these were all counted twice a week. This is Julie showing that she is management material, telling the other students what they should be doing. These are the sites that she visited to do this survey in Niagara, and we also did 4 in Prince Edward County and 10 in Lake Erie North Shore in Essex County. To determine the species, representative samples were collected using fine brushes into ethanol and submitted to the Canadian National Collection of Insects and Arachnids and Nematodes for identification, just using morphology. This shows the distribution of, or the incidence of mealybug and scale at the different vineyards that we visited, and you can see in Niagara that it's predominantly mealybug, grape mealybug, that is present here. A few sites had high numbers of scale, but for the most part, it's mealybug. In Essex County, a little bit more toward the scale side, but pretty small sample size. A lot of zeros. The identifications that came back, all of the mealybugs that were sent were grape mealybug. All of the scale that were sent were European fruit licanium scale. The other thing that was part of Julie's thesis was this intensive monitoring. So twice a week, she went to 4 vineyards. At each vineyard, she would look at 10 vines, do the counts. So 10 vines per site, check weekly from May 14th to August 24th. Same recording numbers of each growth stage on each vine, and we also monitored temperature using a data logger. One of the recommended ways of monitoring for mealybug is to use pheromones. So we purchased pheromone traps and a trace lure, and we also tried monitoring crawlers by wrapping electrical tape backwards, sticky side out, and spraying it with tanglefoot at the bases of canes. Didn't work. Mealybug 0, midges 1,000,000. This is an example of one of the development of the population throughout the growing season of one of the sites that Julie monitored. In blue are the egg clusters. In red are the first instar nymphs, so the ones that are the best carriers of the virus, the ones that are most mobile. In green, second instars. In purple, third instars. This is adults. Eggs are somewhere else, I don't know. Eggs are very low. So you can see that the female peaked just before bloom. The number of females peaked just before bloom. The number of first instar nymphs peaked right around cluster close. But once again, these were primarily found, 99.9% of these were found under the bark. The other thing that we decided to do in 2014 was to look for or determine whether we had vectors of red blotch. So in 6 of the vineyards that were identified in 2013 as having more than 50% of the panels positive with red blotch were monitored. We put out 5 yellow sticky cards per vineyard and took them in every week, changed them. And then we did sweet nut collections in the vineyard and the headlands in late July. So this is me showing that I'm management material because I'm not the one with the sweep nets. Here's Lauren sweeping in the cover crops below the vines and Christelle sweeping in the headlands. I labeled. The species that Christelle has identified out of the thousands that she's looked at, grape leafhopper, aster leafhopper, this one, potato leafhopper, and black-faced leafhopper. She's been testing these for the presence of red blotch, and so far she hasn't found any positives. Good news. Management options, there are no resistant varieties. As I said, some grape varieties will be infected but not necessarily show symptoms, may not have the Brix reduction that is reported in ones that are symptomatic. Very important to plant virus-tested, clean stock, and to rogue infected vines or remove infected vineyard blocks, depending on the degree of infection. So the group from New York did a survey or an evaluation looking at the economic impact of leaf roll virus on Cab Franc. So they sent out a survey to 10 vertically integrated vineyard winery operations that had vineyards with a history of grape leaf roll disease. Within the survey, they documented, the managers reported back on the percentage of the vineyard that was affected, the varieties that were affected, the percent yield loss that they had experienced, and the percent quality reduction. So the yield loss and the quality reduction were used to calculate an economic penalty, and then the cost of removal and replacement vines was also included in the calculation. And from this, a decision matrix was developed where if there were less than 30% yield loss but no penalty, 25% infection and no penalty, just remove the infected vines. Even with a 10% penalty, just remove the infected vines. If there's no penalty, not necessary, or replace. With 30% and 30% yield loss, a little bit more rigorous. When you get to 50% yield loss, you're talking about removing entire blocks. So the economics, the reduction in yield, the reduction in quality balances out with the cost of replacing the vines. So based on this line is 25%. Kind of scary. Here it is in the other red varieties, and here it is in the whites that we sampled. So pretty big impact. Some management options, selective harvest. Some growers I know we're going out and removing the less ripened clusters even more excessively than they would normally do when we got past veraison. There is some work that shows that adjusting crop loads, so doing crop dropping early as opposed to waiting until after veraison may have an effect, may allow the vine to recover to a certain extent, and then managing the vectors of leaf roll. So applying insecticides at the appropriate time. The products that are registered for grape mealybug in Canada, malathion and Movento. We do have dormant oil that's used for mealybug and scale and tree fruit. However, it's not labeled for use on grapes. This is some work that was done by Loeb and Fuchs in New York, where they applied dormant oil or mid-season assail, a neonicotinoid, for crawlers. So oil didn't do much as far as mealybugs were concerned. Assail alone, mid-season, seemed to knock it down. Adding the oil to assail, or doing oil followed by assail, didn't seem to do much. So dormant oil doesn't control mealybugs. Dormant oil works by smothering. The mealybugs are under the bark. So unless you're actually hosing down individual vines, it's very, very difficult to get water and oil down underneath the bark to be able to smother those mealybugs. Assail was only moderately effective, and the timing is really tricky because once again, assail is a contact. It isn't systemic, so it has to come in contact with the crawlers. The crawlers are underneath. A very small proportion are going to be exposed at any one time during the growing season, so your ability to actually affect mealybugs using a contact is going to be relatively limited. They also worked with applying Movento pre-bloom. So here in the blue are untreated vines versus vines that were treated with Movento. In 2011, significant reduction. 2012, the following year, they couldn't find any. So pretty encouraging. They also looked at leaf roll infections, And with Movento applied pre-bloom, the number of newly infected vines was significantly less when Movento was applied pre-bloom. So Movento, effective in controlling mealybugs. It's legal. Excellent control, but it's slow acting. It's also expensive. Sorry, Bear. In order to make it worthwhile, you have to have enough leaf area to have it absorb from the leaves and translocate down into the phloem where the mealybugs are feeding underneath the bark. So our recommendation, apply Movento just before bloom and about 30 days afterwards. This matches with the phylloxera application timing, and the rate is, there's a range of rates. I've recommended the high one, but I'm not the one paying for the spray. So what's next? I'd like to explore the early crop load adjustment and early leaf removal. I have an undergrad student that will be working on that this year. Also looking at asymptomatic infections and their effects on yield and quality. If we can't see symptoms, do we still have that reduction in yield and the reduction in bricks? Want to look at some potential products for vector management. John Mullenhuis and I, with Omafro, will be looking at using the Grape cost of production, which is being produced with 2014 numbers, to do an economic analysis comparable to what Adela and the group in New York did. Researchers at Guelph and Ag Canada are working with me to try to find cost-effective detection methods for grape viruses. It's very expensive to test, to have the samples submitted to the lab for analysis. And then a longer-term project, not just mine, but across Canada, is to develop certified virus-free nursery material. So as I mentioned, the Grape Growers of Ontario, through an Ontario Farm Innovation Program grant, supported a lot of this research. Ontario Grape and Wine Research Inc. also provided funding, and Omafra and Ag Canada provided supplies and the place to work. Couldn't do this without the grower cooperators. I've appreciated their help very much and hope to work with them in the future. And the source of my various PowerPoints that weren't my work, but I'm using with credit, Mark Fuchs, Tom Lurie, Greg Loeb, and Monica Cooper. This is my crew at the beginning of the day before they started sampling. Look at how happy they are. Thank you. Are there any questions? Ed.
For, let's say, Pinot and Chardonnay that are infected, is there a start date for when the reduction in bricks start? Because the plan is to use it for sparkling wine, and if they're still not in balance with the other vines that are unaffected, makes it difficult for harvesting and knowing what the crop is.
You can only do that by hand, right? I'm not sure I understand what you're...
Well, on that chart that showed at 16 bricks, it was already below.
Right.
So is there a date to which the reduction in ripening begins? Is that brazen, like that chart started at, or is it right at bloom? It's a reduction in maturing right from the start of fruit cycle?
I only know from Brazen on. That's all that's ever been tested, so I don't know. Caitlin.
We have an online viewer who's wondering, other than a CFIA survey finding 60% of Ontario blocks had leaf roll, what were the historical surveys that you referred to at the start of the presentation that leaf roll wasn't in Ontario?
I didn't say leaf roll wasn't in Ontario. I said that leaf roll was present in very low amounts, not to the extent. The point is that it was sporadic in vineyards before 2013. We would see symptoms, but in 2013, something happened that it was like an explosion. That vineyards where we knew that it was, almost all the vines could have been infected, showing symptoms. In vineyards where growers might have thought, we had a little bit of problem with ripening the crop in the past, then the symptoms were showing up. So before 2013, we knew it was here. We just didn't realize that the infection was as widespread. And
Again, the lady from California said they didn't think it was rootstock. Possibility to have rootstocks infected and then have disease showing up within or outside of 3 years. Now, I have plantings of stuff that is different clones, and some clones have almost complete infection, and clones right beside it have none, and they were all planted the same year. So is it like it took 20 years for those to start showing symptoms?
that's the other thing that's really different from what's been reported elsewhere. I saw blocks that were planted first leaf that were widely infected. I saw nursery plants this big, that were showing symptoms. And I saw vineyards that were 20 years old, 30 years old, that had never shown symptoms before, just started showing symptoms in 2013. So
The lady from California suggested
Monica.
you had mealybugs came in and infected your vineyard, and now you have it. It wasn't a rootstock or those late infections, a rootstock or a, what do you call it? The material wasn't infected when you got it. It's that you got infected.
The distribution of symptoms that we saw in the vineyard, if you think about this block, it historically had a lot of leaf roll symptoms, but not like this. The mealybugs don't move that fast, they don't move that far. So if it were strictly mealybugs are feeding, remember, it takes at least a year from when the mealybugs feed to when symptoms are expressed, and that's from Monica's work. And they showed up the next year, those were young vines. So it may take longer for older vines to show symptoms. Don't know. The thing is, these diseases are behaving in a way different here than anyone else has reported. And I can't find anyone who can tell me why all of a sudden we had this epidemic in 2013.
Straight after a very hot vintage, wasn't it?
It was a heavy crop year. 2013 was a very heavy crop year. We had excellent conditions for fruit set the previous year, so very heavy crop set. It was kind of cool, a little bit rainy. There was a kind of off-the-cuff comment that cool conditions favored virus symptom development, warm conditions not so much. They have it in California, so I don't know. But yeah, it could be crop load, could be any combination of things. And even in 2014, the degree of symptoms that we saw, the expression, wasn't like what it was in 2013. So it was like we had the perfect storm in 2013 for symptom expression.
Oh my.
Thanks.
Thank you very much, Wendy. That was extremely interesting. Thank you so much. I want to, well, anyway, join me in thanking Wendy. I have a small gift for you. No bugs attached. Well, it is.
That we know of.
A USB
Oh,
stick.
thank
And
you.
thank you very, very much. This, as I said, is our last gathering for 2015. We've had great attendance and great to see everybody online joining us as well. So I wanted to thank everyone for coming, and I wanted to thank Caitlin Little, our communications officer, for coordinating the lecture series. So thank you, Caitlin. So we look forward to 2016, and until then, good luck with all those bugs. Oh my!